Okay, let's begin. Rosalind Allen is our next speaker. About inherent variability in, I can't pronounce that word, amyloid formation. So this shows my knowledge of things. So thank you, Rosalind, for coming. Take it away.
So I'm going to talk about a project that we've been doing, which is about amyloid fibril formation. I'm going to explain what they are in a second. This work is the work of a very talented theoretical postdoc, Uri Savitz-Nothan, who did a project jointly supervised by me and also my colleague Martin Evans here, who's a theoretical physicist, and we also had input from a PhD student, Kim Eden, who did a lot of computer simulations that I briefly mentioned in the talk. But I should also mention that this is a collaboration with my colleague Kate McPhee here, who's an experimentalist, and she actually has a whole experimental program about the kinetics of amyloid fibril formation. And so we learned a lot from working with her and her postdoc Ryan and postdoc Lean and PhD student Ryan. So it's really a joint effort, this project. So as promised, what are these amyloid fibrils? So I guess some of you probably know that a biological cell, a large fraction of its mass is actually made of protein molecules. And protein molecules do all kinds of different tasks inside the cell. So some of them, for example, catalyze chemical transformations. Some of them form actual physical structures that allow the cell to maintain its integrity or to move. So you have all kinds of different proteins that do all kinds of essential things. And the important point about a protein molecule is its three-dimensional folded shape is really important in how it works. And so a protein molecule is made out of a polymer chain of amino acid monomers, and it is designed so that it actually automatically folds into a certain 3D structure, which then allows it to do its task. And that's how proteins are supposed to work. But sometimes it can happen that a protein molecule misfolds into the wrong 3D structure. And it turns out that the incorrectly folded 3D structures sometimes can polymerize into these fibrils, which are held together by hydrophobic interactions, because when the protein chain misfolds, when the amino acid chain misfolds, it quite often exposes hydrophobic patches, which then allow protein monomers to stick together in a way that they're not supposed to do. And these amyloid fibrils are the result of that. So these are actually the polymers which are formed of these misfolded proteins. And this is important because it happens in diseases. So a lot of neurodegenerative diseases are associated with the formation of these amyloid fibrils. And so people would like to understand what are actually the mechanisms of formation and how can you, in principle, stop it from happening. And so there's a whole body of work which is trying to understand this. And that's also what we're trying to do. So one question that we're trying to ask is, can you understand the kinetics of how these fibrils form using simple models, the kind of models that you could maybe solve analytically? And in particular, in this project, what we've been thinking about is how important is molecular noise? So how stochastic is this process of amyloid fibril formation? So how does the kinetics actually work, and what do you actually do when you do experiments? So for example, my colleague Kate McPhee and her students, what they mainly do is this kind of measurement. So they have a plastic plate like this, which is about 10 centimeters in size, and it contains 96 little chambers, which are about 100 microliters or 200 microliters each. And what you do is you put your protein solution in one of these into these little chambers, and you add a fluorescent dye which binds to the misfolded protein. So the amount of fluorescence is going to tell you how much misfolded protein there is. And then what you do is you put this plate into this machine here, which is called a plate reader, and it incubates the plate at a fixed temperature, and it shakes it from time to time. And every 10 minutes or something, it measures the fluorescence in every single one of these 96 wells. So this is a way that you can do multiple replicate experiments quite easily. And what you find out if you do that is that typically the percentage of, so the fluorescence basically, as a function of time, looks like this. So you typically have a lag time, which can be several hours, where you don't detect any fluorescence. And then there's a rapid growth, a sort of a sigmoidal increase in the fluorescence, and then eventually you run out of protein material and the thing saturates. So this project is really focusing on this lag time. So one thing to mention about this is that these experiments, which are typically done, are actually done in quite large volumes. So that the typical volume of one of these experiments is about 100 microliters, which is 10 to the minus 4 liters. But these neurodegenerative diseases, the formation of the fibrils happens in human cells, which is typically a much smaller volume of about 10 to the minus 12 liters. So you can ask the question, well, one thing that people notice about these diseases is that the time it takes before you actually notice symptoms is very variable between patients, and no one knows why that is. And so it's kind of relevant to ask the question, how variable should this amyloid fibril formation process be if you take account of molecular noise? But that's not something that you're going to detect, in these experiments, because the volume here is much larger than it is in a human cell. So what kind of models do people actually make for this process? So actually the models which are thought to be relevant for this are actually quite simple. So typically a model of amyloid fibril formation has 3 steps. So you say that the first thing that happens is nucleation. So there's a waiting time, and then spontaneously a fibril of a critical size, NC, forms spontaneously by some kind of activated process, and nobody knows what that process is at all. And it's very difficult to study because you can't actually see this nucleus forming. So anyway, this nucleus forms by some kind of nucleation process, and then what happens is the fibrils grow by attachment of monomers to the end, like this, and there's some rate constant for that. And what's also believed to happen is that the fibrils can fragment. So once you have a fibril of a certain length, it can break into parts. And that's very relevant because it explains this rapid growth, because it's an autocatalytic process. So when a fibril fragments, it makes 2 new ends, and those ends can then grow. So that the fact that you have this fragmentation makes the process autocatalytic, and that explains why it has this sigmoidal shape. So what's been done quite a lot in recent years in the literature, and also somewhat by us, is the writing down of deterministic description of this model. So you can write down an equation for the dynamics of the concentration of fibrils of length j. So this is actually a whole set of equations, one for each fibril length j, and they're all coupled. And then you've got an equation for the addition of monomers at the end, or terms for the addition of monomers at the end, and they depend on the monomer concentration. And you've got terms for fragmentation, and you've got a term for the nucleation. And it turns out that under some circumstances, when you make various approximations, you can actually solve this equation, and that gives you a deterministic prediction for the kinetics of growth of the fibril mass, so what you measure in this plate reader experiment, and also to some extent for the length distribution of the fibrils, which you can also measure in electron microscopy. And so a lot of what's been done is trying to fit experimental data to the predictions of models like this. And then you can say, well, if my data fits a model with fragmentation, then that probably means that fragmentation is what's happening, whereas if my data fits a model which doesn't have fragmentation but has some other process like end joining or something, then that must be what's happening for that particular protein. So that's actually been very productive. But it doesn't tell you anything about how stochastic this process is, because this is a deterministic description. So there has been some experimental work on what happens in small volumes, but not very much. So this is basically the only study which has been done at least recently on this, and that was by Thomas Knowles and his collaborators in 2011. And what they did is actually make microfluidic droplets, which are about 10 to the minus 10 liters in size, and they track the formation of amyloid fibrils in these droplets under the microscope. And they discovered some interesting things. So one thing that they discover, which is, it was actually that what happens in these droplets is that a fibril forms at a random time, the first fibril forms, and then it spreads in space. So they actually track the spreading of these spatial waves of fibril formation, which previously nobody had known that they existed. What we're going to focus on in this talk is that they also were able to observe the individual stochastic nucleation events in different droplets. And from that they could measure the lag times in these individual droplets, and so that gives some information about the stochasticity of this process. But they only did 52 droplets, so the statistics were not quite what one might have wished. But they did discover some interesting things about this lag time, and the main thing that they discovered is that the lag time, if you plot it on a graph versus 1 over the volume of these droplets, so the 52 droplets they had were all slightly different volumes, and so you can make a plot of the inverse of the volume of the droplet versus the lag time. And it turns out that if you do that, what you find is that it looks like this, And so you can write the lag time as an equation with two terms in it. One of the terms depends inversely on the volume, and the other one is a constant with respect to the volume, and that's this offset here. And so that presents a picture of the lag time where it's actually got two components. So they say that there's a nucleation time, which is the waiting time to the formation of the first nucleus, and that scales inversely with the volume, as you would expect for a kind of Poisson nucleation process. But then there's also a growth time, which is the time that it takes you to actually detect the existence of this growing fibril, and the growth time accounts for this offset here, and that's what they found out. So what we wanted to do is to actually try and make a theory to account for the variability in this lag time, and in particular the variability in the growth time, and also in the nucleation time to the formation of the first nucleus. And we started off by thinking about some of our own data from Kate's experiments, which are for bovine insulin. And for bovine insulin, the fibrillation kinetics is well described by this fragmentation model, and you can see that here. So this is some of our data from a large volume experiment in this plate reader device. And here's the black is the data, and the red is a fit to the deterministic model that I described before. So it fits really well. And then what you can do is you can say, well, seeing as we know these parameters for example, the elongation rate and the fragmentation rate from the deterministic fit, why don't we just do a Monte Carlo simulation, but in a much smaller volume? So we take the same parameters, but we reduce the volume to 10 to the minus 12 liters, which is what we think is a human cell volume. And then, and that's what our student Kim did. And you can see that indeed the simulation predicts that there should be significant variability in the lag time. And interestingly enough, it's only the lag time which is variable, so the growth rate of these curves is pretty much non-variable. So it seems like the lag time is the interesting thing to study for this problem. So the question then is, can we predict the distribution of these lag times? So the first thing you could do is you could write down a stochastic version of the deterministic of the full model. So we could say, well, let's track the number of fibrils ni of length I. And so the state of the system should be described by all of these integers, which are the numbers of fibrils of different lengths. And we could write a master equation for the probability of being in a particular system state. And so the processes which should be in there are basically the same processes as I described before. So we have nucleation at some rate alpha, which is a function of the monomer concentration, which now I call C of t. And that should depend. So I define this parameter epsilon, which is a volume scaling. So epsilon is 1 over v times Avogadro's number, and so this is basically a conversion factor. It converts concentrations into molecule numbers. Okay, and then we should have growth at the ends of the fibrils, and we should have fragmentation. Okay, and so you can write down this stochastic master equation, and it's got terms for nucleation, and it's got terms for growth at the fibril ends. And it's also got a lot of terms for fragmentation, which are too small to even read. And so in conclusion, this is a complicated master equation, and maybe some people here could solve it, but we certainly were not able to make any progress with this master equation. And so, okay, so much for the full model. So what we decided to do instead was to actually coarse grain the model. And so rather than tracking the whole fibril length distribution, which is what makes this model complicated, instead we decided to track only two things. So the total number of fibrils, which you can get by summing over the numbers of all of the fibrils of different lengths, and also the total number of monomers in the fibrils, which is proportional to the mass, the total mass of the fibrils, which you can get by summing over the length times the number of fibrils of each length. And so if you think about it, that reduces the problem quite a lot, because you've only got 3 possible transitions. So you've got nucleation. What nucleation does is it increases the number of fibrils by 1, and it increases the number of monomers in fibrils by the nucleus size nc, and it's got some rate associated with it. And then you've got growth of fibrils at the ends, and what that does is it doesn't change the number of fibrils, but it increases the total fibril mass by 1 unit. And the rate of that actually depends on the number of fibrils, n, because each fibril has a chance to grow, so the more you've got, the more you can grow. And then you've got fragmentation. So fragmentation doesn't change the mass of the fibrils, but it increases the number of fibrils by 1. And the rate of that depends on the total fibril mass, because we assume that longer fibrils have more chance to break. They've got more sites where they can break. So you've got a process that increases m by 1 that depends on n, and you've got a process that increases n by 1 that depends on m. So it's kind of an interesting set of coupled equations that we end up with. Yeah?
The last one, if it is proportional to the average length of the fibril, shouldn't it be m divided by n?
No, it's not proportional to the average length, it's proportional to the total length. Because the number of places that you can fragment.
And everything, right?
Yeah. And another thing to mention is that because we're talking about the lag time where the monomer concentration is basically not much depleted, we decided to say that the monomer concentration is just going to be the total monomer concentration, because that makes it a lot easier to solve the equations, and it should be okay because the total mass of fibrils is anyway small in the lag time, during the lag phase. So that what you can then do is you can sum this horrendous master equation over like this to get a master equation in the variables n and m. And so you've got the master equation describes the probability of having n and m at time t, and it turns out to look like this. So it's a lot smaller than the other one, and it's got terms for nucleation, it's got a term for growth at the fibril ends, and it's got a term for fragmentation. And we've redefined some variables. So now mu describes the growth at the fibril ends, and lambda now describes the fragmentation rate. I wouldn't have done this, but Uri likes to do this kind of thing. So the aim is to solve this master equation to find the probability distribution for the lag time, and we have to define what the lag time actually means. So what we say is that the lag time is going to be the time for this fibril mass to reach a predefined threshold, which we call mt. So how are we going to do this? So what we decided to do is to do a linear noise approximation, which is also known as Van Kampen's system size expansion. And what you do there is, or probably you already know this, but anyway, what you do is you assume that these variables can be decomposed into a deterministic part, which is here, and a stochastic part which scales like 1 over the root of the volume. So the variance in the stochastic part is assumed to scale like 1 over the volume. And this is a very standard thing to do. So the deterministic part describes the dynamics of the mean of these two variables, whereas this part is going to describe fluctuations around the mean. And is this a good approximation to do? Well, it may or may not be. So what this approximation requires is that the fluctuations in these variables are actually small compared to the mean. And you could say, well, that's not so good for the lag time, because actually the mean is very small. And that's true. So what we expect is that this approximation should be okay for the growth part of the lag time once it's actually got started, but it probably isn't okay for the nucleation part. So we'll see about that. So I'm going to briefly sort of work through how you do this system size expansion, but I don't want to kind of dwell on the details too much. Firstly, the deterministic part. So this is just the dynamics of the mean, and here we write it in terms of the concentrations of these two variables. And so the reason we've got this is to convert concentrations into molecule numbers. So these are molecule numbers and these are concentrations. And you can write down deterministic equations for these two concentrations. So for example, the concentration of fibrils increases due to fragmentation with the fragmentation rate lambda. So this is the concentration of monomers in fibrils. And it also increases due to nucleation. And then the concentration of monomers in fibrils increases due to fibrils growing at the ends, and that's proportional to the actual number of fibrils. And it also increases due to nucleation. And so this is kind of a nice set of equations because psi depends on phi and phi depends on psi. And it turns out you can solve those ones and you get this. And then you can then say, well, let's find out the lag time by setting the concentration of fibrils equal to the threshold and finding out what t is. And if you do that, you get this, where we've defined some more variables, but these ones don't really matter. So the only thing that really matters in this expression for the lag time is actually the prefactor, because everything else is inside the log, so we don't mind about it. This doesn't really have much effect. And what's interesting about this prefactor is that it says that the lag time, firstly, it scales as the square root of two parameters multiplied by each other, the inverse square root, which are the elongation rate and the fragmentation rate. And perhaps more relevantly for experimentalists, it scales as the inverse square root of the monomer concentration, because this parameter mu is actually proportional to the monomer concentration. And this is not new, actually. It's already emerged from solutions of the deterministic equations by other people, in particular Thomas Knowles and his group. And so that's nice because it actually is correct. And it's also relevant because it turns out that what people often measure in these experiments are the scaling of the lag time with the monomer concentration. And this 1 over square root dependence is a very characteristic dependence. And so if you see that, then you kind of know that it's a fragmentation model that is appropriate. So it's kind of nice to see that. Okay, but then what about the fluctuation? What about the stochastic part? So now we have to consider these stochastic variables which describe the fluctuations around the mean. And their probability density of these stochastic variables x1 and x2 obeys a Fokker-Planck equation, and you can derive the coefficients, the diffusion matrix and the drift vector, in this Fokker-Planck equation by doing a Taylor expansion of the stochastic master equation, and that's what Uri did. And it turns out you get these things, and you can actually solve the Fokker-Planck equation to get an expression for p of x and t, which has a sort of Gaussian form. I didn't write down the full thing here because it's a bit long. But anyway, since you know this p of x and t, you can actually find the lag time distribution. And so, as I said, the lag time is the time when the number of monomers in fibrils exceeds this threshold. And how do you calculate that from this Fokker-Planck type description? What you have to do is a first passage time calculation. So you want to know the first time at which the numbers of monomers, the distribution of the first time when the number of monomers in fibrils exceeds this threshold. And the way that you do this is you actually think about the probability that the number of monomers in fibrils is higher than the threshold, and that's just a quantity. And then you can say, well, how does that change in time? So if you consider a time t to t plus dt, how does the probability of being above the threshold change? Well, it changes by an amount that equals the lag time, because the only way you can cross the threshold is by actually exiting the lag phase. And so it turns out that the time derivative of this probability is actually the lag time distribution. And that's a standard thing in first passage time statistics. And so if you can calculate this probability and take its time derivative, that tells you the lag time distribution. What I've kind of glossed over is the fact that we have this denominator, and the reason for this is because if you did a full stochastic simulation, you actually would always eventually form fibrils, right? But because we've done this linear noise approximation, actually the dynamics sometimes hit zero in a very small probability, in a very small fraction of cases, and we actually normalize by that. So here we normalize by the probability that you actually end up with fibrils at the end. And this is just a fudge, really, but it turns out to be only a small fudge. Okay, so this thing, the probability that m is bigger than mt at time t, you can compute it from this probability distribution. And so here's what we do here. So rather than integrating, we've transformed an integral over n and m into an integral over x1 and x2. And then you can just do this integral. And it turns out that this is the answer. So the lag time distribution is this, and it's written in terms of a different variable to make it look simpler. But what you can then do, what's interesting about this is to compute its moments. So fortunately, as you would expect, the mean of this lag time distribution agrees with the deterministic prediction. So that's encouraging. And then the standard deviation of it is given by this. Here I'm kind of cheating because actually I made some assumptions, which is that the fragmentation is slower than the elongation, but it turns out that that's actually true. Otherwise, you get a very complicated expression. But if you make this assumption, you get a very simple expression here. And this is sort of interesting because what it says is the variance, which is sigma squared, scales like the mean in terms of the parameters. So we remember that the mean lag time scaled as 1 over the square root of mu times lambda, and so does sigma squared here. But it has a different dependence on the protein, on the monomer concentration, because it's got this factor of alpha to the half, or alpha for the sigma squared, and alpha has is a nucleation rate, and it has its own dependence on the protein concentration. So what we find out is that the variance of the lag time scales differently with the protein concentration compared to the mean of the lag time. And actually it decreases faster as the monomer concentration increases, because alpha is an increasing function of the monomer concentration. So is this actually correct, you could say? Well, how can we test it? So what we can do is compare the predictions for this lag time distribution with the kinetic Monte Carlo simulations of the full stochastic model with all of these different fibril lengths. And that's what our student Kim did. And the answer, and here are the results for 3 different nucleation rates alpha. And you can see that it does work quite well, but as the nucleation rate decreases, it works less well. And that's kind of what we expect, because the nucleation rate is controlling the time to formation of the first nucleus, so the nucleation time. And we expect that this approximation is not going to work so well when that's actually significant. So it gets worse, and that's what we expect. So what do we do in the case where the actual waiting time to the first nucleus is long, and so this Van Kampen expression doesn't work very well? Well, actually, it's quite simple what you can do there. So we assume, as everyone else assumes, that this nucleation process is a one-step Poisson process. So you have a waiting time, which has a known distribution, and it's this. So it's an exponential distribution, and the rate parameter is the nucleation time divided by this volume scaling thing. Okay, and so we know the waiting time for this nucleation event, and we can just include that in the prediction of the lag time distribution. So what we say is, we're going to write a probability distribution for this two-step process, and we know the probability distribution for this, and now we've calculated the waiting time distribution for the second thing from our Van Kampen expansion. And so what we're going to do is a convolution of the probability distribution for this and the probability distribution for the lag time, but in that calculation of the Van Kampen thing, we have to set the initial conditions to be one nucleus at time 0, because that's what you get out of this Poisson process. So if you do that, you get another expression for the lag time distribution, and it turns out that that one actually works even when the nucleation time is slow. So here are some results with a much slower nucleation time, and now it works. And so that gives new predictions for the mean lag time, which is then the Poisson lag time mean plus the Van Kampen lag time mean, and also for the standard deviation of the lag time, which is the standard deviation. This is not obvious, but it turns out to be true. Standard deviation of the Poisson process plus the standard deviation of the growth process. And what's also interesting is that these two contributions to the variance actually scale differently with the protein concentration. So I don't know if you remember that, but this one was proportional to 1 over alpha, alpha being the nucleation time, which depends on the protein concentration, and this thing is proportional to 1 over alpha squared. So as the protein concentration increases, this term becomes more and more important, the variability that comes from the growth process. So one thing you can ask is, does this prediction actually agree with what experimental data exists, which is these 52 droplets by Thomas Knowles. And 52 droplets is not very many droplets, so it's not really, because all the droplets all have different volumes, it's not really possible to plot a lag time distribution because you've only got 1 or 2 droplets in each volume bin. But what you can plot is little dots for each of Thomas Knowles' droplets. And what's good about this is that the parameters for the model are actually extracted directly by Thomas Knowles from his measurements. So we don't have to do any parameter fitting here. We just take the parameters that he's measured and plug them into our model. And the green shows what we predict for the mean lag time and what we predict for the standard deviation shown by the error bars. And what you can see is it's at least not inconsistent with the experimental data. And seeing as it's not fitted, that's actually sort of good. And what we also find by looking at this data, you can't see it here, but we can see it from the maths, is that in this regime, actually, it's the nucleation time that dominates the lag time. And the time to growth, which we put all this effort into computing from our Van Kampen expansion, actually doesn't really matter for the variance in this regime. But if we had a higher protein concentration, which still could be in the physiological range, actually it would matter. So it was still good that we computed it. So that kind of brings me almost to the end. So from the theory, what we conclude is that this molecular noise in amyloid fibril formation can matter when protein formation happens in small volumes like the volume of a human cell, and this might be relevant, who knows. And we also find out that it's got these two components. So the variability has a component which is the time to form the first nucleus, and another component which is the time before you can actually detect formation of the fibrils, and both of them have got simple scaling with the parameters, but it's actually different scaling. In particular, they scale differently with the protein concentration. And what we also conclude is that these 52 droplets are just not enough droplets to make a test with experiments. And so in Edinburgh, we're actually trying to develop ways of doing this more systematically, doing experiments more systematically. And so my colleague Simon Titmuss in Edinburgh is actually trying to develop a microfluidic device which will make thousands and thousands of little droplets in which we could grow amyloid fibrils and measure the statistics. And here's a picture of some preliminary results. What this shows is many, many little droplets which have been made by this device. And here are some other preliminary results in a slightly different device where you can actually see fluorescence coming from amyloid fibrils that have been formed. So eventually, hopefully, we can do more statistics. So that's the end, and thanks for your attention. Yeah?
So when you add the nucleation time to the lag time, was that assumed to be independent
Yes.
of everything else? Okay, so...
What, you mean the lag time and the nucleation time are assumed to be independent of each other?
Yeah, is every...
Yes.
Yeah. Okay, so does that not explain the standard deviation result? That
Oh, yeah, you're right. It does.
Oh, okay.
Yes, sorry. Yes,
Okay.
it does. Yes.
What did you mean by kinetic Monte Carlo simulation?
Gillespie algorithm.
Of the reduced model, right? The one with M and N.
No, it wasn't the reduced model. It was the full model, I think, wasn't it?
Okay.
Yeah, it's the full model.
Okay.
Yeah, with all the length distribution in it.
Okay. But do you lose anything going to just M and N? It didn't look like it, right?
Well, what you're doing, in making the transformation, you sum over the master equation.
Yes.
So I think that means that your description should be correct as long as you're only looking at M and N.
Yeah.
So yeah, you don't.
But
You lose
need
information
more
about the fibril length distribution.
But you need that. Okay. I see.
Well, some people would say you would like to know that because some people believe that the actual toxic agents in these diseases are the small fibrils,
Right.
and so it's kind of nice to know how many small fibrils there are, which you
Okay.
don't know if you do this.
Yeah.
But on the other hand, we couldn't solve it if we didn't do this. So. Yeah, at the back.
Yeah, I was a little confused when you said this is autocatalytic. It seemed like the reaction you chose was a polymer of length n is consumed, as is a monomer, and a polymer of length n+1 appears. So where's the catalysis there? I didn't.
Well, that one isn't autocatalytic. So that's
Well-
what you're talking about is the elongation reaction, and that isn't
Right.
autocatalytic. But what I'm talking about is the fragmentation reaction. So there you have a fibril which splits into two fibrils.
Both strictly smaller than the original.
Both strictly, yeah. You're not creating any mass by doing that. But because you've got more ends, and the ends are the things that grow, you've actually created more sites where the fibril can grow. So if you combine that with the growth reaction, then the whole thing is autocatalytic. Because the longer you get, the more places you can split.
Okay, so like if you think of ends as a species, those once you get lots of long things, then the ends.
Yeah.
Okay.
Yeah. Yeah.
But I was kind of curious if the deterministic model could explain the scaling between the lag time and 1 over volume. I know you were focusing on trying to explain the variability at a given volume, but that scaling, if you just took this model and just scaled the volume down, it would give you that. Yeah, I don't know, on the previous slide.
Well, the deterministic model. Oh, that's not it.
Yeah, that scaling.
This one. So it has a nucleation term in it, and this will be volume dependent because the. Will it be? No, it won't. It won't, because the deterministic equation only talks about protein concentrations, right? This is the concentration of fibrils, and these are the concentration of monomers. So I think it has no volume.
But surely the concentration depends on the volume. So if you just increase, well, I just don't know how these droplets were made. Or
Well, if you just increase the volume without increasing the fibril number, then sure. But that's not what you do.
Okay.
You keep the concentration fixed and you decrease the volume.
Oh, I see. So in those droplets, the concentration is the same.
Yeah.
Ah, right. Okay.
Yeah. So I think the deterministic model won't tell you anything about that.
Right. Yeah.
That's right.
Is it obvious that the microscopic reverse process of the fragmentation, sort of the joining, is unimportant?
No, it's not obvious. Actually, a referee asked that, and so and you can put it in, but of course it depends what rate you choose. What rate you choose, and so we put it in. And if you put in a sufficiently small rate, then and you can calculate what the rate should be from the thermodynamics. And so it seems like it doesn't matter in this case. But there are other models where you have a much more rapid end joining process, and that seems to apply to other proteins. Like we studied this protein called ovalbumin, and that gives a completely different kinetics. So it seems like for some proteins it matters, but not for bovine insulin.
Just related to that question, actually, you could seed your system so that you can play with the lag time, right? Information, and that gives you different kinetics. And have you done the experiments for that kind of?
We haven't done seeding experiments, but lots of people have done seeding experiments. And what it does is basically abolish the lag time. But it's true that if you put in sufficiently small number of seeds, maybe you could still get the lag time. Yeah, it's a good idea. I don't know if anyone's thought about that specifically. Yeah.
So
Yeah, this is going to the physiological thing that you had on your last slide. So I guess the question is that we know a little bit about the amyloid fibrils that cause disease. We also probably have enough patient data to look at the variation in time to the development of the disease. And I was just curious if have you tried to look at that, whether or not something like this, say choosing a good sort of mass of fibrils that is going to be equivalent to having those small toxic species, do you get anything that's even remotely close to the kinds of scaling that we
I
have?
have no idea.
in humans?
We haven't done it. I would doubt it, to be quite honest.
Yeah.
But no, we haven't. It would be kind of nice to look, but I would be astonished if this really.
Yeah,
is
I would, but it would be astonishingly cool. So it might.
Yeah.
Because maybe it does. Maybe it just falls out.
Yeah, maybe.
So you may have mentioned this, but under, let's say, these biologically relevant conditions, I guess the time is determined by the nucleation time, or is still the actual growth important there?
Well, it depends what protein concentration you put in, but it seems more likely to be the nucleation time. But the biological cells have all kinds of other mechanisms for removing misfolded protein and stuff like that. So I think this model is rather simplistic for an actual biological cell.
More questions? Let's thank Rosalind again for a great talk.